Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start a topic, please like, share, and subscribe the channel. In the last session, we dealt with isoenzymes. In today's session, we will continue the same. Identification of isoenzymes. Certain points are mentioned regarding the same. First point is that in agar gel or polyacrylamide gel electrophoresis, the isoenzymes have different mobility. The LDH, CK, and ALP isoenzymes can be separated by electrophoresis. Then third point is heat stability. One of the isoenzymes may be easily denatured by heat. Example, bone isoenzyme of ALP. Fourth point is inhibitors. One of the isoenzymes may be sensitive to one inhibitor. Example, tartrate labile ACP. Next point is Km value or substrate specificity may be different for isoenzymes. Example, glucokinase has high Am, whereas hexokinase has low Km for glucose. The next point is cofactor requirements may be different for isoenzymes. Next point is mitochondrial isocitrate dehydrogenase is NAD plus dependent, whereas cytoplasmic isoenzyme is NADP plus independent. Next point is tissue localization may be different for isoenzymes. Next point is H4 form of LDH is present in heart, while M4 variety is seen in skeletal muscle. Specific antibodies may identify different types of isoenzymes. For example, CK isoenzymes are separated by isoenzymes. Coming to the next chapter 6, chemistry of carbohydrates. Functions of carbohydrates. Carbohydrates are the main source of energy in the body. Brain cells and RBCs are almost wholly dependent on carbohydrates as the energy source. Energy production from carbohydrates will be 4 kilocalories per gram. Storage form. Carbohydrate act as storage form of energy. For example, starch and glycogen. Excess carbohydrate is converted to fat. Glycoproteins and glycolipids are components of cell membrane and receptors. Carbohydrates act as structural basis of many organisms like cellulose of plants, exoskeleton of insects, cell wall of microorganisms, and mucopolysaccharides as ground substance in higher organisms. The general molecular formula for carbohydrate is CnH2n. For example, glucose has a molecular formula Ca6H12O6. Carbohydrates are polyhydroxyaldehydes or ketones or compounds which yield these on hydrolysis. Coming to the next point, nomenclature. Molecules have only one actual or potential sugar group and are called as monosaccharides. In the Greek language, mono is nothing but one and saccharide means sugar. They cannot be further hydrolyzed into smaller units. When two monosaccharides are combined together with elimination of water molecule, it is called as disaccharide. Trisaccharides contain three sugar groups. Further addition of sugar groups will correspondingly produce tetrasaccharides, pentasaccharides, and so on, commonly known as oligosaccharide. In Greek language, oligo means a few. When more than 10 sugar units are combined, they are generally named as polysaccharides. Polysaccharides having only one type of monosaccharide units are called homopolysaccharides, whereas those having different monosaccharide units are heteropolysaccharides. Sugar having aldehyde groups are called aldoses, whereas sugars with keto group are called ketoses. Depending on the number of carbon atoms, the monosaccharides are named as triose containing C3, tetrose containing C4, pentose containing C5, hexose containing C6, and heptose containing C7, and so on. Here are some examples of monosaccharides based on their carbon atoms. If the number of carbon atoms is 3, then the generic name should be triose, and glyceraldehyde is an aldose, whereas dihydroxyacetone is a ketose. Number of carbon atom is 4, then their generic name should be tetrose, and erythrose is a aldose, and erythrulose is a ketose. If the number of carbon atoms is 5, then the generic name should be pentose, and arabinose, xylulose, ribose are aldose, whereas xylulose and ribulose are ketose. If the number of carbon atoms is 6, then the generic name should be hexose, and glucose, galactose, mannose are aldose, whereas fructose is a ketose. If the carbon number of carbon atoms is 7, then the generic name should be heptose, and pseudoheptulose is a ketose. In this diagram, we can see that keto group and aldehyde group of a, of a sugar changes. If R1 is present, it is ketone, whereas if H is present, it is aldehyde. Coming to the next point, stereoisomers. Compound having same structural formula but differing in spatial configuration are known as stereoisomers. While writing the molecular formula of monosaccharides, the spatial arrangements of H and OH group are important since they contain asymmetric carbon atoms. Asymmetric carbon means that 4 different groups are attached to the same carbon. The reference molecule is glyceraldehyde or glycerose, which has a single asymmetric carbon atom. In this diagram, we can see that D-glyceraldehyde and L-glyceraldehyde are mirror images of each other. The number of possible stereoisomers depends on the number of asymmetric carbon atoms. It is given by the formula 2 raised to n, where n is the number of asymmetric carbon atoms. The configuration of H and OH groups at the end at the second carbon atom of glyceraldehyde may be noticed. The two mirror forms are denoted as D and L varieties. All monosaccharides can be considered as molecules derived from glyceraldehyde by successive addition of carbon atoms. Therefore, penultimate carbon atom is the reference carbon atom for naming the mirror images. This is also referred to as absolute configuration. Here in this diagram we can see D-glucose and L-glucose, where OH group as is attached to the fifth carbon is on the right side in case of D-glucose, but on L-glucose it is located on left side. With a reference to the penultimate carbon atom, that is C5 in the case of glucose, the configuration of H and OH groups is changed and two mirror images are produced. It may be noted that D and L varieties, the groups in second, third, fourth and fifth carbon atoms are totally reversed so as to produce the mirror images. These two forms are also stereoisomers. These sugars are naturally occurring sugars, and body can metabolize only these sugars. Coming to the next point, optical activity. In the presence of asymmetrical carbon atom causes optical activity. When a beam of plain polarized light is passed through a solution of carbohydrates, it will rotate the light either to the right or to left. Please note that D and L notation has no bearing with the optical activity. Depending on the rotation, the molecules are called extrorotatory, that is plus and D, or levorotatory minus L. Thus, D-glucose is dextrorotatory, but D-fructose is levorotatory. Equimolecular of an equivolecular mixture of optical isomers has no net rotation. That is why it is called racemic mixture. Coming to the next point, diastereomers of glucose. Configurational changes with regard to C2, C3 and C4 will produce 8 different monosaccharides. Out of these, only 3 are seen in human body. They are glucose, galactose and mannose. In this diagram, we can see that hexose is of physical importance. D-glucose is a blood sugar is in blood sugar and main source of energy in the body. D-fructose is a constituent of sucrose and common sugar. D-glucose constituent of lactose, glycolipids and glycoproteins. D-mannose is a constituent of globulins, mucoproteins and glycoproteins. Here we can see that D-glucose, D-mannose and D-galactose are epimers of D-glucose. There are 8 diastereomers isomers of aldohexoses. With reference to C5, all of them have D and L forms. Hence, the molecular formula of hexose, that is C6H12O6, represents 16 different monosaccharides due to spatial arrangement of constituent groups. Glucose is the most predominant sugar in the human body. It is a major source of energy. It is present in blood. D-glucose is dextrorotatory. In clinical practice, it is often called as dextrose. Galactose is a constituent of lactose or milk sugar and glycoproteins. Galactose is epimerized to glucose in liver and utilized as a fuel. The term galactose is derived from Greek word gala, which means milk. Mannose is a constituent of many glycoproteins. Mannose was isolated from plant mannans, hence the name. Here ends our session. Kindly like, share and subscribe the channel. Thank you.